Back when Aristotle was around, people didn't know much about the origins of life. A lot of folks, including the famous Greek philosopher himself, thought that non-living matter could just create life. They called it spontaneous generation, and they used it to explain all sorts of things. Like, if someone was storing grain in a silo and happened to find mice that hadn't been there the day before, well, they assumed the mice arose spontaneously from the non-living bricks, mud, and some bits of grain. Aristotle even suggested that semen had a unique property that allowed it to enliven the menstrual blood. That's how babies were made. And sure, it's closer to the truth than the unified stork theory that your parents came up with, but still definitely wrong. These days, we know that life doesn't arise spontaneously from inorganic material. It's actually made of tiny, individual building blocks called cells. But it took a lot of science to get there. Science that relied on all the work that came before it. Hi, I'm Dr. Sammy, your friendly neighborhood entomologist, and this is Crash Course Biology. Now ready your mind for one of the most complex theme songs ever constructed. We actually knew about the cell a few hundred years before spontaneous generation had been debunked, thanks to a physicist named Robert Hooke. In 1665, around the same time that Isaac Newton was thinking about gravitational forces, Hooke was focused on science at a smaller scale, a microscopic scale, actually. Having made some tweaks to an existing microscope, Hooke discovered something astonishing. Looking at a slice of cork under his improved scope, he was amazed by the tiny pores he saw. To him, they looked like little rooms in monasteries that monks lived in, which are called cells. And, well, the name stuck. If Aristotle had a microscope, we probably wouldn't have gotten hung up on spontaneous generation for so long. But that's how scientific advancement works. It's all about having the right tools for the task and being able to iterate or build onto the work of past scientists. In the mid-1800s, Hooke's sketches of the microscopic world led a couple of other scientists, physiologist Dr. Theodor Schwann and botanist Matthias Schleiden, to propose that all organisms are made from cells, and that the cell is the basic building block of life. Then, in 1855, the work continued when Dr. Rudolf Virchow added his own proposal. All cells come from pre-existing cells that have multiplied. These three ideas became the cornerstones of what we now call classical cell theory. It wasn't until the 1900s that we figured out the key differences between the two major cell types, prokaryotic and eukaryotic, thanks in part to more advanced microscopes. You're probably more familiar with the eukaryotic variety because those are the cells that make up most of the living things that you see every day, like bees, trees, and people. And unless you're using a microscope, you're unlikely to see any prokaryotes like E. coli, a type of bacterium that can cause some nasty infections. Although we humans do owe a lot to prokaryotes since they form itty-bitty colonies in our guts and on our skin, helping us digest our dinner and even ward off infection. The best evidence so far suggests that single-celled prokaryotes were among the first forms of life, and that our eukaryotic cells evolved from them about 2.7 billion years ago. So they're like your great, you get it, you're related. There are a few important differences between the two types of cells. Our eukaryotic cells have a defined nucleus, usually near the center of the cell. Prokaryotic cells don't. In fact, their name means pre-nucleus. The nucleus is where the genetic material is stored in a eukaryote, packed up neatly within the nuclear membrane, a double-layered shell that surrounds the nucleus. Most prokaryotes have their single circular piece of DNA just kind of hanging out in the main compartment of the cell with everything else in a water-based jelly called cytoplasm. So prokaryotic cells kind of look like they packed their suitcase in a hurry, while eukaryotes made sure to bubble wrap their cellular accessories and tuck them away in special compartments. This compartmentalization lets eukaryotes develop more complex, coordinated cellular processes than prokaryotes. Like, take plants, for example. Not only do eukaryotic plant cells run some complex processes, but they also do some extra work to support the unique structure of plant life. For starters, much like the buffalo wings I had for lunch, plants are boneless. To make up for that, each cell membrane is braced by a surrounding cell wall that helps plants maintain their structure. This thick barrier consists of structural molecules, including carbohydrates and proteins. Plants also have a large central vacuole, or cavity, that stores a lot of water and chemicals that the plant needs. The central vacuole also provides additional structural support alongside the cell wall. And then there's the chloroplast, which converts sunlight into an energy that the plants can use. Both a plant's vacuole and its chloroplast are membrane-enclosed structures called organelles. In the same way that our heart pumps blood and our lungs exchange gases, these little mini organs inside a cell each serve their own unique function. In a way, a cell is like a city, with each organelle performing its own civic duty to keep the city cell working in a coordinated way. Let's say goodbye to the plant cells for now and go on a thought bubble tour of the eukaryotic animal cell. Welcome to Cellular City. Here at the entrance to the city, we find a barrier of biomolecules called lipids surrounding the cell. The barrier is the cell membrane. It's studded with proteins, some of which open doors allowing us access. We have a very efficient transportation department here in Cellular City. The highways are made up of protein filaments of the cytoskeleton. They help move cargo and let the cell maintain or change its shape as needed. They're quite dynamic too. See how they assemble at one end while disassembling on the other? As we approach the center of the cell, you'll catch a glimpse of City Hall, the nucleus. Here, important genetic messages get sent out in the form of the nucleic acid RNA. Please, no flash photography. Once the instructions reach the ribosomes over in the cytoplasm, they turn the instructions from City Hall into a protein that helps make action happen in the body. Now, if you look to your right, you'll see that sometimes the nucleus dumps the RNA right into these buildings of the endomembrane system, where they enter the endoplasmic reticulum. We just call it the ER around here. And it has two sections, the rough and the smooth. The rough ER, as you can see, is dotted with ribosomes of its own. So it makes proteins same as the cytoplasm. But here in the ER, the cell can make more complex proteins with modifications. The smooth ER is the lipid manufacturing plant, where new pieces of the cell membrane are made, along with those famous message-carrying lipids called hormones. Don't worry, they'll be around at the end of the tour for autographs. These cellular products are shipped out of the ER in a vesicle, a fluid-filled structure that buds from the smooth ER's lipid membrane carrying cargo to the rest of the cell. Those vesicles need to make a quick pit stop at the Golgi apparatus, part manufacturing plant, part protein sorting facility. The Golgi is another member of the endomembrane system, which packages proteins into vesicles like this one. And finally, we end our tour at the mitochondria, the city's power plants, where energy for all of life's cellular processes is produced by breaking down the right molecules at the right times. All of this while making versatile chemicals that can be used as building blocks in other areas of the city. And that ends our tour for today. Bye-bye now. Bye-bye. Bye-bye now. Bye-bye. Thanks, Thought Bubble. We gotta give that tour guide a raise. They were great. Anyway, how amazing that so many complicated things are happening inside a single microscopic cell. You might have noticed that our tour of the animal cell didn't include a stopover in the chloroplast. That's because the chloroplast is unique to plants. While animals don't have chloroplasts, plants do have mitochondria, double the power production for our leafy friends. I'm not jealous. Okay, I'm a little jealous. So while we non-plants might have chloroplast envy, both of these organelles are really special and really strange when you consider their origin. One day, 1.5 billion years or so ago, we think a small bacterium found its way inside of a larger bacterium. Whether on purpose or by accident, who can say? But this new living arrangement actually worked well for both bacteria. These days, we call this arrangement endosymbiosis. Both mitochondria and chloroplasts might have arose from this kind of symbiotic relationship that worked so well that it became permanent, and it allowed them to co-evolve into the eukaryotic cells that we know today. In other words, we think that that's what led to the cells that allow bugs, bananas, and bears to exist. Scientists had long believed that this could be the case, but it wasn't until the 1960s that the idea of endosymbiosis as the origin for eukaryotic cells really took off, thanks to evolutionary biologist and zoologist Dr. Lynn Margulis. Margulis, drawing on the work of many scientists before her, hypothesized that both mitochondria and chloroplasts were descended from remnants of the prokaryotic cells that had merged in an endosymbiotic relationship. But what she had that earlier scientists didn't were the right tools. Thanks to advancements in microscope technology which helped her present experimental evidence to back up the hypothesis. Further proof that science doesn't happen in a void. It builds and grows and changes as different people pick it up across generations. People who are in the right place at the right time with the right tools. And speaking of continuing the work that came before you, remember classical cell theory? Well, we call it classical for a reason. Today we have modern cell theory. Modern cell theory builds on its classical counterpart in the way that Margulis built on the work of her predecessors by adding 3 more central ideas. Energy flows within cells, similar species have similar cells, and cells divide and pass along their genetic information to new cells. And just like classical cell theory wasn't possible without the right advancements in science, neither was modern cell theory, which also relied on advancements in microscopy. We've come a long way since the old-school microscopes of Robert Hooke's day. Of course, the reason it takes such powerful microscopes to observe cells is because the vast majority of cells are really, really small. As cell size increases, the volume inside the cell increases faster than the surface area along the membrane, and the surface-to-volume ratio puts a limit on cell size. If a cell got too big, there wouldn't be enough membrane to support all of its processes. That's why we're not made up of just a couple really big cells. Some cells dodge the size limit by changing their shape, taking the form of rods or spiky balls that increase their surface-to-volume ratio. Like Caulerpa taxifolia, a single-celled organism that can grow larger than a human arm. It's plant-shaped with these cool fronds that increase the surface area. There's also more than one nucleus to support this giant water-dwelling thing. So even though most cells are really small, nature is always full of surprises. From Hooke to Margulis to, well, us, the history of the cell is a long chain of scientific iteration and experimentation. As far back as the world of ancient Greek philosophy, folks were asking one of the most important questions in science. It's a simple question, only 4 words long, but it's one that can help us understand cell theory, invent new microscopes, and figure out new ways of thinking. Why did that happen? By asking that simple question, we've been able to discover not only the existence of the cell and its major types, but we've also learned about the busy, bustling internal structures of organelles, and hypothesized how cells adapted over billions of years to become the shapes of life that we see today. Next time, we'll check out the cattywampus membrane that holds them all together. I'll see you then. This series was produced in collaboration with HHMI BioInteractive. If you're an educator, visit biointeractive.org/crash course for classroom resources and professional development related to the topics covered in this course. Thanks for watching this episode of Crash Course Biology, which was filmed at our studio in Indianapolis, Indiana, and was made with the help of all of these nice people. If you want to help keep Crash Course free for everyone, forever, you can join our community on Patreon.